Hello, my name is Professor Hudson. Let us continue with the narrated PowerPoint lectures for Chapter 9 that cover mitosis and cancer. In the last segment, we were talking about how abnormal regulation of cell cycle genes can lead to cancer. In the following slides, you will see some review questions based on the previous narrated PowerPoint lecture. Please record the letter for the correct answer for the next set of questions. Question 13. When mutations occur in proto-oncogenes, they become cancer-causing agents called. Choose the correct answer. Oncogenes, fill in the blank. Choose the correct answer. This is question 14. The cell cycle in cancer. Let's talk about some other reasons that cancer may originate besides mutations in the proto-oncogenes and tumor suppressor genes. One theory about cancer and how it originates has to do with the telomeres. Chromosomes normally have special material at each end called telomeres. These get shorter with each cell division. When they get very short, the cell will no longer divide. Telomerase is an enzyme that maintains the length of telomeres. Mutations in Telomerase gene cause telomeres to continue to lengthen, which allows cancer cells to continually divide. So normally, the shortening of the telomeres helps the cell to know, one, its age and how many divisions it's been through, and when it's time to stop dividing. Mutations in the telomerase enzyme will cause the telomeres to stay long or lengthen, which throws off this regulation, and the cells will continue to divide. Let's look at a slide that discusses how lung cancer develops. As we stated before, all cancers begin with a single cell, one of trillions of cells, that mutates in certain key genes. As the number of mutations accumulate, the benign tumor can go and become a malignant growth. And then metastasis can occur when additional mutations develop. During metastasis, cells from the tumor break off and enter the bloodstream or surrounding body fluids. This slide also shows a picture of a normal lung and a cancerous lung. Note the cancerous lung has a large tumor. Also, the cancerous lung is brownish and black. This lung was taken from a smoker. Breast cancer develops the same way. Once again, a single cell out of trillions of cells develops a mutation in certain key genes. This causes the cells to grow uncontrollably into a tumor. The tumor can be benign and stay in its original location, or additional mutations can develop, allowing the tumor the ability to invade neighboring tissue. Additional tumors can develop on top of those, which allows the tumor cells to break away and enter the lymphatics or the bloodstream. Cancer cells will spread through the lymph and blood vessels to other parts of the body and set up tumors in alternative locations. This slide just summarizes some differences between normal cells and cancer cells that we discussed earlier. The last segment of our narrated PowerPoint lectures deals with prevention, diagnosis, and treatment of cancer. Let us begin. Cancer can be prevented, or at least the risk of developing cancer can be lowered. Frequent screenings can help detect cancer when it's in its early stages or detect benign tumors before they become malignant. Eating a diet low in fat and high in fiber can also help reduce the risk of cancer. Eating more vegetables and less animal products helps lower your risk for cancer. Exercise lowers your risk for cancer. Studies have shown that if you exercise at least 30 minutes a day, every day, you lower the chances of developing cancer. Also, you should avoid known carcinogens. And a carcinogen is something that causes cancer. Known carcinogens are UV sunlight, X-rays, certain viruses, and tobacco smoke. How cancer is diagnosed. Once again, screening tests help to diagnose cancer. The more frequently the screening tests are done, the earlier the cancer can be detected. Early detection increases the odds of surviving cancer. Pap smears are used to detect cancer of the cervix. Mammograms are used to detect breast cancer. Colonoscopy is used to detect tumors and cancers in the colon. Scientists are trying to develop tumor marker tests where they can look for specific proteins in the cells that would alert them to ones that are cancerous. Also, there are genetic tests now that can identify certain genes that increase the odds of you developing cancer. For example, they have identified a breast cancer gene. Carrying this gene or 2 copies of it significantly increases your risk of developing cancer of the breast. If something is noted during the screening, the doctor must confirm the diagnosis. Usually a biopsy is done where a small sample of the cells is removed from the lump or the tumor and examined under the microscope. Pathologists usually handle this, or the doctor, and they will look for certain key things in order to determine whether the tumor is benign or malignant. Ultrasound can be used to detect some tumors and radioactive scans. If cancer is found, several treatment options are available. Radiation therapy, where radiation is used to target the specific cancer cells. This disrupts the process of cell division. Chemotherapy involves intake of certain drugs and chemicals that specifically stop cell division in rapidly dividing cells. Most of the cells that are targeted would be the cancer cells since they are rapidly dividing. However, normal cells that rapidly divide, such as these cells that produce hair and the lining of the digestive tract, will also be disrupted. Immunotherapy is a relatively new strategy where scientists hope to somehow trigger your own immune system to kill the cancer. Excision is commonly used, especially with benign tumors or ones that have not invaded or become malignant. In excision, basically the tumor is removed or cut out from where it is. The thing to be careful with excision is that if you leave even one cancerous cell behind, the tumor can reappear. Another strategy is to block angiogenesis. Angiogenesis is where new blood vessels form. Cancer cells will often secrete chemicals that cause blood vessels to form when they weren't there before, or to increase in number, thereby feeding the tumor. If you can block the formation of these blood vessels, you can starve the tumor cells and they will die. Let us review. Question 15. New methods of cancer treatment prevent blank, which cuts off the supply of nutrients and oxygen to the tumor. Choose the correct answer. Question 16. The prevention of cancer may be accomplished by. Choose the correct answer. This concludes the narrated PowerPoint lecture for Chapter 9. Note, there were not 20 questions this time. For the narrated PowerPoint lectures for Chapter 9, when you take the lecture quiz, you should have answers for 16 questions. Note, you will need the letter that corresponds to the correct answer for each question. You will input the letter only in the PowerPoint lecture quiz.